In connective tissue, the architectural and mechanical identity is determined by what lies between those cells, the extracellular matrix. The extracellular matrix is divided into an aqueous ground substance and a structural network of protein fibers. This highly hydrated ground substance acts as a medium for diffusion, exchanging nutrients with the blood supply. Suspended within is the solid fiber scaffold. The stroma serves as a meticulously engineered, load-bearing matrix. That scaffold is built from three main fibrous components: collagen, reticular fibers, and elastic fibers. This histological slide shows type 1 collagen, the body's primary tension-resisting fiber. Found in bone, skin, and tendon, these thick bundles are highly acidophilic, staining a distinct pink with eosin. Under an electron microscope, a precise structural pattern emerges. Adjacent pro-collagen subunits overlap each other by exactly 67 nanometers, creating the alternating dark and light longitudinal striations characteristic of type 1 fibrils. This chart categorizes the wider collagen family. Type 1 forms thick, tension-resisting fibers, while type 2 provides resistance to pressure in cartilage without forming large bundles. Further down, type 4 collagen acts as a sheet-forming network, supporting epithelial basal laminae. Type 3 collagen, which forms reticular fibers, requires a different approach. Being heavily glycosylated, they are argyrophilic, appearing as a delicate black lattice network only when impregnated with silver salts, as seen in this lymph node. Finally, expansible organs like the lungs and large arteries require elastic fibers. Like reticular fibers, elastin has poor H&E affinity and requires specific stains like orcine, which reveals the dark branching network you see in this spread preparation. Recognizing these specific fiber types and their staining affinities is the only way to reverse engineer a tissue's mechanical limits directly from a histological slide. All connective tissue originates from embryonic mesenchyme. This mesoderm-derived tissue is packed with a viscous ground substance and lacks heavy fibers, leaving these undifferentiated, spindle-shaped cells scattered throughout the matrix. As the tissue matures, it requires an architect. The fibroblast descends directly from mesenchyme, taking up permanent residence in the tissue to manufacture the extracellular matrix. You can determine a cell's current workload by its histology. The active fibroblasts here display large, euchromatic nuclei and abundant cytoplasm filled with rough endoplasmic reticulum. Nearby, quiescent fibrocytes are smaller, with dark, heavily condensed heterochromatic nuclei. The active fibroblast's physical appearance is dictated by its function. Its massive cytoplasm is essentially a factory floor dedicated to mass manufacturing protein. Collagen synthesis is a rigorous multi-stage assembly line split across two environments, the intracellular factory and the extracellular assembly zone. This schematic maps the entire pathway. The process begins inside the cell, where mRNA is translated into pro-collagen alpha chains in the rough endoplasmic reticulum. Moving down, specific residues undergo hydroxylation, an enzymatic reaction requiring vitamin C as a cofactor. Next, select hydroxylysine residues are glycosylated. 3 alpha chains are aligned and stabilized into a tightly wound triple helix before being packaged into transfer vesicles. Through exocytosis, these vesicles fuse with the cell membrane, ejecting the intact pro-collagen molecule into the extracellular space. Outside the cell, the first extracellular modification occurs. Pro-collagen peptidases cleave the terminal, non-helical peptide sequences from the ends, transforming the water-soluble pro-collagen into an insoluble collagen molecule. Faster, more energetic pace. Once cleaved, these rigid, rod-like protein subunits spontaneously self-assemble in an entropy-driven process, locking together end-to-end to form thick, striated fibrils. Looking at the bottom of the schematic, the final stabilization step takes place. An enzyme called lysyl oxidase creates covalent crosslinks between the adjacent molecules, locking the fiber's structural integrity into place. Because collagen is engineered across 2 biological domains, a failure at any single molecular checkpoint compromises the structural integrity of the entire organ. While fibroblasts build the matrix, a different class of cells uses it as a defensive battlefield. These are the sentinels, leukocyte-derived cells that migrate from the bone marrow to patrol the connective tissue. This electron micrograph reveals a macrophage, a highly phagocytic cell derived from circulating monocytes. Notice the highly irregular, pleated surface structure with deep indentations. and the abundance of dense secondary lysosomes required for digesting extracellular debris and presenting antigens. Depending on where these monocytes settle, they take on specific regional names. This table details how they adapt, becoming Kupffer cells in the liver, microglial cells in the central nervous system, or osteoclasts in bone. Plasma cells are another resident sentinel. They are identified by a spherical, eccentric nucleus with clumped peripheral chromatin resembling a clock face. The pale area adjacent to the nucleus is a massive Golgi apparatus required to terminally glycosylate the antibodies they secrete. Positioned strategically near small blood vessels, mast cells are packed with bioactive substances like histamine and heparin. In this light micrograph, the extreme density of these dark, basophilic secretory granules almost completely obscures the central nucleus. These granules are the payload for type 1 hypersensitivity, or anaphylaxis. This diagram illustrates the mechanism. Primary exposure to an allergen causes B cells to produce IgE antibodies, which bind to receptors on the mast cell membrane. Upon secondary exposure, the green antigens cross-link those specific IgE receptors. This immediately triggers massive calcium entry, leading to the rapid fusion and exosexosis of the granules, dumping vasoactive mediators directly into the local microenvironment. This extracellular matrix acts as a preloaded biological tripwire, capable of triggering an instant systemic inflammatory cascade. This comparative table highlights how board-relevant connective tissue diseases result from specific failures on the cellular assembly line we just mapped out. Returning to the intracellular factory in the rough ER, scurvy results directly from vitamin C deficiency. Without ascorbic acid to facilitate the hydroxylation of proretin, the triple helix cannot properly stabilize, resulting in widespread tissue breakdown and bleeding. Osteogenesis imperfecta strikes at the very beginning of the process. A genetic failure in type 1 collagen transcription results in defective pro-collagen alpha chains, robbing bones of their tension-resisting scaffold. Ehlers-Danlos syndrome represents a spectrum of assembly errors. Depending on the variant, the failure can range from faulty translation to missing extracellular pro-collagen peptidases, resulting in hypermobile joints and fragile, highly extensible skin. Conversely, keloid formation is a regulatory failure. Fibroblasts ignore normal feedback signals and aggressively oversynthesize type 1 collagen, causing thick, ropey protein fibers to pile up into a dense, disorganized mass. Connective tissue is a meticulously balanced equation of matrix and cell. When you understand the baseline rules of its synthesis, its pathological collapse becomes entirely predictable.